Good evening, viewer. Welcome to our FTV English version. Here's some breaking news. Researchers have discovered a promising new approach to gene therapy by reactivating genes that are normally inactive. Yeah, they achieved this by moving the genes closer to regulatory elements on the DNA known as enhancer. To do so, they used CRISPR-Cas9 technology to cut out the piece of DNA separating the gene from its enhancer. And this method could open up new ways to treat genetic diseases. The team demonstrated its potential in treating sickle cell disease and beta thalassemia, two inherited blood disorders. In these cases, a malfunctioning gene might be bypassed by deactivating an alternative gene that is usually turned off. So, this technique called delete to recruit works by altering the distance between genetic elements without introducing new genes or foreign material. The study was conducted by researchers from the Haverset Institute, DLAT Group, Erasmus MC and Sanquin, and it published in the journal Blood. Genes in our DNA contain the instructions for making proteins, which carry out many essential functions in cells. However, not all genes are active at all times. Some are only switched on when certain nutrients need to be processed. So, while others are active only during early development and later shut down, proper cell function depends on tightly controlled gene activity. One important regulatory mechanism involves enhancers, which are segments of DNA that act like switches to turn genes on. Enhancers can be located right next to the genes they regulate or positioned much further away along the DNA. In this study, we discovered that it is possible to activate a gene by bringing it closer to an enhancer, said Anna Karina Felder, one of the study's first authors. Felder and her colleagues Zjord Jalsama, Han Vegan, and Regine Majet accomplished this using CRISPR-Cas9, a gene editing tool that works like precise molecular scissors. BNS exclusive report, FTV English, Delhi, Bharat.